Hi everyone, we are from Group 2. My name is Anis Maisara. My metric number is E24A3316 and this is my member.
Hi, my name is Shaira Izzati. My metric number is E24A3298. Hi, I am Nur Ain Sophia. My metric number is E24A3290. Hi, my name is Nur Izzati binti Jabir. My metric number is E24A3300. Hi, my name is Nur Aida Safrina binti Syabuddin. My metric number is E24A3309.
Hi, my name is Farsha Afia binti Tajidin. My metric number is E24A3318. Bye!
Assalamualaikum and hi everyone. My name is Mirza Fikra binti Jabri. My metric number is E24A3300. So today I will explain about a number of chromosomes of salmon DNA. So the first is the genus of Onchoryncus encompassing species like a rainbow trout and Chinook salmon exhibit a range of chromosome number and sex determination system. Both males and females within a species typically share the same diploid chromosome number, but variation exists due to chromosomal polymorphism and sex chromosome differentiation. For example, a rainbow trout has commonly 16 number of chromosomes, while Chinook salmon has 68 number of chromosomes. However, both male and female have the same number of chromosomes as long as they are in the same species. For your information, the different number of chromosomes is caused by whole genome duplication, Robertsonian fusion and fission, and others. As you can see, in Diagram 1, the metaphase spread of the China genetic rainbow trout with 58, 59, 60, and 62 chromosomes after DAPI staining A, B, and C, and PNA fish with telomeric probe in D. The arrow show the X chromosome. The next picture is Diagram 2. The chromosomes are labeled 1 until 27, representing the 27 pairs, and each pair consists of 2 chromosomes, one from each parent. For the homologous pair, similarity between chromosomes in each pair indicating they are homologous that carry genes for same trait. If male and female individuals, pair 27, may represent sex chromosome, for example, XX in female and XY in male. Lastly, we have the drawing pictures of the chromosome.
Next, number of genes. The genome of rainbow trout, Onchoryncus mykiss, a salmonid species, is complex due to a whole genome duplication, which is called WGD, event around 80 million years ago. This has resulted in the retention of many duplicated genes, contributing to its adaptive success. According to Berthelot et al. 2014, the rainbow trout genome contains about 46,585 protein coding genes, which is higher than in other theloids. These genes are distributed across 48 to 64 chromosomes comprising metacentric and ecocentric types. The WGD has provided genetic material for traits like growth, disease resistance, and environmental adaptability. Recent chromosome level assemblies confirm this structure and refine our understanding of kilocyte, including those who aquaculture relevant traits. The unusual high number is due to a salmonid-specific whole genome duplication, which is SS4R WGD event, which created many genes duplication called homolog. About half of the rainbow trout's genes are still present as duplicates, while the rest have reverted to single copies, which is singletons. Recent genome assemblies such as the early homozygous line confirm this high gene count and reveal complex duplicated region. The retention of so many homologs is thought to help rainbow trout adapt to different environment, but it also makes genetic studies more challenging.
Salmon DNA in pharmaceutical. Polydeoxyribonucleotide (PDRN) is a DNA derived compound primarily extracted from the sperm of salmon species such as Onchoryncus mykiss, rainbow trout, and Onchoryncus kita, cum salmon. It consists of purified mixture of deoribonucleotide polymers, typically ranging from 50 to 2000 base pair in length. Although PDRN does not function as a gene or protein itself, it plays a significant biological role in promoting tissue repair, regeneration, and anti-inflammatory responses. One of the primary mechanisms through which PDRN acts is by activating the A2A subtype of adenosine receptor, which are known to regulate anti-inflammatory and tissue healing processes in the body. Additionally, PDRN provides a rich source of nucleotide precursors that support DNA synthesis and cellular proliferation, making it particularly effective in wound healing and skin regeneration. This mechanism also promotes the expression of vesicular endothelial growth factor (VEGF), contributing to angiogenesis or the formation of new blood vessels. Salmon DNA is used due to its high structural similarity to human DNA, excellent biocompatibility, and low immunogenicity. Because of these properties, PDRN is widely applied in dermatology, aesthetic medicine, orthopedics, and wound care. Clinically, it is used in products aimed at improving skin texture, reducing signs of aging, repairing scars, and treating chronic wounds such as diabetic foot ulcer. In summary, PDRN from salmon sperm is safe and biologically active compound that facilitates cellular repairs and regeneration by modulating key molecular pathways and supplying essential building blocks for tissue recovery. When PDRN binds to this receptor, it does four important things. First, reduces inflammation. It helps calm down the immune system so that it less redness, swelling, and pain in damaged or irritated areas. Second, promotes tissue repairs. PDRN provides building blocks for DNA, helping cell grow and repair damaged skin or tissues. Third, improve blood flow. Angiogenesis. It increases the production of a natural protein called VEGF, which helps grow next blood vessel. This brings more oxygen and nutrients to the healing area. Last one is boost collagen and fibroblast activities. It activates fibroblasts, the cell that make collagen, which is essential for strong, healthy skin and faster wound healing. In short, PDRN helps the body heal faster, reduce inflammation, grow new blood vessel, and straighten the skin by supporting natural repair processes at the cellular level.
Now, let's continue to the method. First, we have sample collection. Juvenile chum salmon were collected from 3 different locations on the Pacific coast of Iwate Prefecture, northern Japan. We have Miyako Bay, 54 fish, Yamada Bay, 25 fish, and Kamaishi Bay, 10 fish. Then, the throat length of each fish was measured. Their stomachs were then removed and preserved in 99.5% ethanol at negative 30 degrees Celsius. Let's look at the morphological observation of stomach contents. Each stomach was dissected and the prey items were sorted using a stereo microscope. Prey were identified to the lowest possible taxonomic level using a stereo microscope and a light microscope, following the method of Chihara and Murano. The number of prey in each stomach was counted. Even digested or broken prey were identified using key body parts to avoid double counting. The proportion of each prey type was calculated based on the total number of prey. All prey remains were then preserved in 99.5% ethanol at negative 30 degrees Celsius for later analysis. Next, DNA-based analysis of stomach content. The first point is DNA extraction. DNA from the whole prey organisms in one stomach was extracted using the Powerfical DNA isolation kit, following the manufacturer's instruction. To design a PNA clamping primer specific to chum salmon, DNA was also extracted from the muscle tissue of 16 juvenile chum salmon from Miyako Bay using the DN-Easy blood and tissue kit, also following the manufacturer's instructions.
Next is PNA-mediated PCR sampling. Researchers use DNA from cum salmon muscle to study a mitochondrial gene called cytochrome oxidase. oxidase 1, CO1. They use PCR polymerase chain reaction to amplify this gene with two specially designed primers. The PCR process involves heating the DNA at 94 degrees Celsius for 30 seconds, cooling 55 degrees Celsius for 30 seconds to allow primer to attach, and then heating to 72 degrees Celsius for 1 minute to build the DNA. The resulting DNA fragment, about 1970 base pairs long, were cleaned and directly sequenced using a specialized machine. These CO1 gene sequences were then stored in international genetic database. To focus on identifying prey DNA, not the salmon's own DNA, the researchers create a special PNA, peptide nucleic acid primers. These PNA primers block the salmon's CO1 genes from being amplified. This allows only prey DNA from the salmon's stomach contents to be amplified and studied. For high throughput sequencing, the universal CO1 primer will modify these necessary tags and adapters. PCR for this step involves 30 cycles with lower annealing temperature 42 degrees Celsius to help amplify DNA from a wide variety of prey species. After that is CO1 sequence analysis. CO1 amplicons were sequenced using a 454 GSVLX sequencer. Raw text sequences were processed with the pipeline in model 1.31.1 and primer were removed from both ends. Sequences shorter than 300 base pair were excluded. The remaining reads were grouped into OTUS operational taxonomy units based on 98% sequence symmetry using sequencer 5.1 for each O2U. A representative sequence were identified and analyzed using BLASTIN and BLASTX to remove bacterial CO1 and unrelated gene and eliminate duplicates. Finally, the relative abundance of each prey taxon was calculated using model based on the proportion of read.
This slide shows the relative abundance of stomach contents of juvenile chum salmon based on morphological observations and DNA-based analysis. Stomach contents from an individual sample from Miyako Bay. Stomach contents from an individual sample from Kamaishi Bay. Stomach contents of chum salmon observed by microscopy. There's three results found in the observation. First is Tropepid eucalenus bumi, Tropepid neocalenus plum crust, and Amphiophid termisto japonica.
From the method, salmon DNA will be used in agriculture. First method is DNA extraction. The product produced is total genomic DNA from stomach contents. Agriculture or aquaculture use first supports precision aquaculture by tailoring nutrition to growth stage or environmental condition. Second, useful in developing sustainable, cost-effective fish feed alternative. Number 3 is enables monitoring of diet composition in farm fish. For the method number 2 is PNA-mediated PCR clamping. The product produced is enriched prey DNA. Agricultural or aquaculture use enables non-invasive diet analysis in farm, raised or wild animals, fish, poultry, or and livestock. Can be used to assess the impact of different feed regimen on gut content or health. And lastly is applied to trace food source or contaminants in food chain.
Last method, COI sequence analysis, product use, species identification data, barcodes, agricultural or aquaculture use, builds reference database for feed, organism, pests,
or pathogens, support biosurveillance in aquaculture systems, for example, detecting invasive prey or predators. Lastly, it allows verification of species composition in commercial feeds, like ensuring no protected or contaminated species are included.
Next is method for agriculture. Salmon DNA or salmon gene-based product shows emerging potential in agriculture, particularly to enhance plant growth, improve soil quality, and stimulate plants' immunity. The process starts with collecting waste materials from salmon processing industries, such as sperm, gonads, skin, or muscle tissues. These parts are selected because they are rich in nucleic acids, especially DNA. The collected tissue washed and homogenized to prepare for DNA extraction. Cell membrane then will broke down using a combination of chemical detergents like SDS, enzyme, and heat of agitation. Like lysis, organic solvents such as phenol chloroform or silica column-based purification methods are used to isolate and purify the DNA, while other remaining RNA or proteins are removed using RNA assay or protease treatment. Then, the DNA is precipitated using cold ethanol or isopropanol, centrifuged and redissolved in water or a buffer solution. The DNA will hydrolyze into smaller fragments using enzymatic hydrolysis DNAC or similar nucleases to make it more readily usable by plants. The resulting product consists of short DNA particles or free nucleotides which more easily absorbed by plant's root or leaf and can act as metabolic stimulants. Once fragmented, the DNA will be formulated into agriculture products. It can be mixed with liquid carriers such as water, humic acid, or seaweed extract to build foliar sprays or combined with solid carriers like biochar, compost, or peat for soil amendment application. The formulated salmon DNA is applied to test crops such as lettuce, tomatoes, maize, or rice in agricultural settings. The experimental group received different concentration and varying application frequency, while control group receives no treatment or only standard fertilizer. The treatment delivered via foliar spray, soil drenching, or seed coating depends on the trial design. After the application, several parameters are measured to evaluate the effectiveness of the salmon DNA treatment, including seed germination rate, root and shoot length, leaf chlorophyll content, total biomass, and final crop yield. In some cases, plant resilience to environmental stresses such as drought or salinity is also assessed. Lastly, the statistical analysis such as ANOVA or t-tests are used to compare the result of the treatment and control groups. This allows researchers to determine whether the salmon DNA had a statistically significant positive impact on plant growth, yield, or stress resistance.
That's all from us. Thank you. Bye.